Hello, everybody. Welcome. Welcome to the weekly Fralin Biomedical Research Institute Pioneers and Biomedical Research Seminar. Before I turn it over to Dr. Lamouy to introduce today's speaker, Dr. Atfi from VCU and the Massey Cancer Center, just want to mention, and by the way, as you'll learn, I'm sure we have a real French access connection here with today's seminar, so I'll leave that for Sammy to talk about. But just remind everybody that next week, the next installment on January 20th, will be Dr. Mark Andeman from Beth Israel Deaconess Harvard, and that'll be jointly sponsored by the neuroscience group, and Dr. Andeman works on serotonergic gating from the brainstem of retinal flow through the thalamus and the brain. So for the neuroscience folks, that'll be in that area. And I want to just mention again that today's seminar that Sammy will introduce is jointly sponsored by the Cancer Research Group here at the FBRI. So without further ado, Sammy, I turn it over to you.
All right, thank you, Mike. All right, thank you all for coming physically and on Zoom. So it's my great pleasure to introduce today's PBR speaker, Dr. Azeddine Atfi. Dr. Atfi is professor in the Department of Biochemistry and Molecular Biology at VCU in Richmond, and he's also the leader of a new cancer biology program at Massey Cancer Center. And this is a program that includes 45 full members in 15 different departments. Dr. Atfi got his PhD in molecular and cellular biology in at the University of Rennes in France, and then following postdoctoral training at University of McGill in Montreal, Canada, Dr. Atfi then started his own lab at the hospital Saint-Antoine. Hôpital Saint-Antoine, I'm going to say it in French, it's easier. Hôpital Saint-Antoine in Paris, where he became also research director for one research unit at INSERM. So INSERM is the French equivalent of NIH. And so through his PhD postdoc and as a group leader, Dr. Atfi quickly became an expert in the field of TGF-beta signaling in cancer progression, specifically understanding, dissecting transcriptional regulator downstreams of TGF-beta, downstream of SMAD, including TGIF1, TWIST, CJUN, and so of course, that came from that major contributions in the field of TGF-beta that led to many publications in top journals, including EMBO Journal, Molecular Cells, Cancer Cells, Cell Reports, and Nature Cell Biology. And so this is when Dr. Atfi was in France that I met him. Because I was working on TGF-beta signaling during my PhD, I asked Dr. Atfi if he wanted to join my thesis committee, and he graciously accepted. Although Dr. Atfi was in Paris and I was in Grenoble in the Alps, it's quite far from each other. We didn't have Zoom at that time, and so I just realized, it's funny, I just realized that now that Dr. Atfi is in Richmond, we're much closer here in the US than we were when we were in France, which is surprising because France is a quite small country. So Dr. Atfi then moved in the US in 2008 after a detour in Boston. He became professor at the university, in the Department of Biochemistry at the University of Mississippi Medical Center in Jackson, before then joining VCU in 2018. And so I just want to highlight one field that I found really important, interesting, fascinating that Dr. Atfi and his group has been working on recently. And I think that he's going to mention that toward the end of his talk is cancer cachexia. So cancer cachexia is a loss of muscle mass that happens in cancer patients. It decreases, of course, the quality of life, survival for patients. And so there is this fantastic discovery that Dr. Atfi and his team made, was published 4 years ago in Developmental Cells, where they found that TWIST1 is a regulator of protein degradation in the muscles, and that causes cancer cachexia in the context of pancreatic cancer. All right, so throughout his career, Dr. Atfi received many awards and honors in France, Canada, and in the US, with most recently in 2020 receiving the Mary Anderson Harrison Distinguished Professor in Cancer Research Award. And his research is currently funded by 3 R01 from NCI. And I want to mention also that Dr. Atfi is a permanent member of the NIH study section, TPM, tumor progression and metastasis. So Today, Dr. Atfi is going to talk about genetic drivers in pancreatic cancer. Please join me in welcoming Dr. Atfi. Thank you.
Yeah. Thank you very much, Sammy. Can you hear me? Yeah. So when I met yesterday, Sammy, I was surprised, okay, and pleased to see him. He's not changed, okay, really. And he said the same to me, but I said to him, yes, but with a lot of more gray hair, okay, but I was pleased to see him. And so I am delighted to be here today and to share with you my work over the years on TGF-beta and more recently on pancreatic cancer. And later during my talk, I will share with you some results on cachexia as well. So I am interested in TGF-beta since I started my career almost 25 years ago. And so the problem working with TGF-beta, it has many, many functions during tumor progression. And sometimes it gets very complex and even like confusing. So anyway, so TGF-beta inhibits proliferation of most epithelial cells and most of cancer cells. They escape the growth inhibitory response to TGF-beta, which supports the notion that TGF-beta is a tumor suppressor gene in cancer. And so the mechanism that accounts for this loss of the tumor growth inhibitor response to TGF-beta can be mediated. I'm sorry. I have to start with this. Can be mediated through inactivation of the receptor, inactivation of downstream effectors like SMED proteins, and activation of oncogenic signaling pathway like KRAS, which is mutated up to 90% in patients that they have pancreatic cancer. So, and at a late stage of tumor progression, you can see here that now TGF-beta can act as a tumor promoter and induces invasion and metastases. And you see here the mechanism. It induces angiogenesis and epithelial to mesenchymal differentiation, and also it regulates tumor microenvironment. Just to give you a quick overview how TGF-beta works, it binds initially to this receptor, is a transmembrane serine kinase receptor. And when it binds to this receptor, T-beta-R2, it recruits this second receptor, T-beta-R1. And this leads to the formation of this complex between T-beta-R2 and T-beta-R1. T-beta-R2 has a constitutive kinase activity, and it phosphorylates the T-beta-R1. And this leads to the activation of the kinase activity of T-beta-R1. And then the activated kinase of this receptor phosphorylates two proteins, SMED2 and SMED3. And you can see here that the phosphorylation of these two proteins is facilitated by these two proteins, SERA and CPMA. And once it gets activated, T-beta-R1 phosphorylate. This leads to the dissociation between SMED2 and SMED3 and SERA and CPMA. And then you can see that SMED2 and SMED3 associate with SMED4, another SMED protein, and translocates to the nucleus where they regulate the expression of TGF-beta target genes. So, and in the nucleus, SMEDs can activate or repress transcription. And this is mediated by binding directly to DNA or binding to DNA to interaction with other binding factors such as FAST1. And so to activate transcription, you can see SMED binds to CBP and P300 to acetyltransferases that facilitates transcription by acetylating chromatin and opening chromatin. And to repress transcription, you can see that SMADs can bind to this transcriptional co-repressor TGF1, which recruits a complex containing HDAC, and this leads to transcriptional repression of TGF-beta target genes. So, so this is that just I showed to you. TGF binds to SMAD and records HDAC and represses transcription. And this is the first mechanism published by a big group led by Dr. Massey in New York, and the work was published in Cell in 1999. And a postdoc in my lab in 2002, she wanted to run a control experiment, and she used in that experiment a truncated form of TGF that we call TGF delta SBD. She confirmed that this truncated form of TGF cannot bind SMAD2 in the absence or presence of TGF-beta. And she found that the wild-type TGF can associate with SMAD2. And this association is increased by TGF-beta. So everything is fine. The controls, everything is fine. So the problem that when she used this truncated form of TGF delta SBD, she saw that it can still inhibit TGF-beta dependent transcription. This is the wild type, this is the mutant, exactly with like similar efficiency, suggesting that TGF might not inhibit TGF-beta signaling as reported by the Massey group in Cell. And we believe that this is the right mechanisms by which TGF inhibits TGF-beta signaling without associating with SMADs. So, and to understand this phenomenon, so we decided a long time ago to perform a yeast-to hybrid screening to identify TGF partners that might contribute to its inhibitory function in TGF-beta signaling. You can see in this table, we identified many partners. We published many great papers with this screening. And in red, you see here CPML and C-Jun as interacting factors for TGF1. CPML, this is the one that we talked about when we talked about the phosphorylation of SMAD2 by the activated TGF-beta receptor. So we did a whole bunch of experiments. And to make this long story short, to what we saw is that TGF can inhibit the phosphorylation of SMAD2, which is the first step of SMAD signaling. And you can see in this co-immunoprecipitation assay, you can see that TGF interact with CPML in wild-type cells, but this interaction does not exist in cells deleted for C-Jun, indicating that C-Jun mediates the interaction between TGF and CPML. And the mechanism that we came up with is this one, is that TGF interacts with CPML via C-Jun, and this leads to sequester CPML into the nucleus away from the cytoplasm, where it binds to sera and facilitates phosphorylation of SMAD2 by the receptor. And this is how TGF interrupts TGF-beta signaling. So the other partner that we identified in this screen is axin-2 that you can see here in red. And axin-2 is, as you know, is an inhibitor of TGF-beta signaling. So here again, very quickly, so we did a whole bunch of experiments to summarize. So we found again that TGF sequesters axin into the nucleus and thereby stabilizing beta-catenin. And this leads to activation of Wnt signaling. So in this graph, you can see we used wild-type cells and cells deleted of TGF. And when you delete TGF, Wnt cannot induce anymore the expression of its target genes, cyclin D1, CMYC, and LEF1. And you see that TGF is itself activated by Wnt3, indicating that TGF is maybe functions in a positive feed-forward loop to activate Wnt signaling. This is a very simple but robust experiment in vivo. So if you delete TGF in this transgenic mouse model, these mice, they do not develop at all mammary tumors, suggesting that TGF plays an oncogenic role in breast cancer. So this is where, the conclusion that we had about TGF when we did this in vivo experiment. But we have also a TGF mice with a global knockout of TGF in the whole body. So, and we maintained colonies of these mice in the lab. And after 12 months, 14 months exactly, we found that some mice start to develop several types of cancers, like this one and this one, okay? This one is huge. And so we were able to characterize these tumors as lymphomas and liposarcomas. So lymphomas, we think it is B-cell lymphoma because you can see they are positive for the B-cell marker P220. And liposarcoma, we did not performed any analysis, but it looks like the human well-differentiated liposarcoma. So here we are in a situation that TGF functions as a tumor suppressor gene. So we are confused, okay, in the mammary gland, it is an oncogene, and here it is a tumor suppressor gene. So we do not want to continue this game analyzing TGF in different types of cancer. And so we decided just to go ahead and to study the role of TGF in a global manner in all tissues. And to do that, so we use this system so that we call the Sleeping Beauty system. And this is, in my opinion, is the most powerful tool to study genetic alterations in cancer. So please let me explain. So Sleeping Beauty mice, you see that they have two transgenes. This one has several copies of a transposon ONC3, and the second transgene has a transposase that is under the CMV, ubiquitous CMV promoter. And between CMV and transposase, you have a LOXP stop LOXP cassette that prevents the expression of transposase when CRE is not there. And when CRE is there, what happens is that this, I'm sorry. Yeah, so this LOXP stop LOXP cassette is excised out, and this allows CMV to drive the expression of transposase. And then transposase will cleave and release several, I would say, thousands of copies of the transposon that moves around and integrate the genome in a random way. Creating alterations. For example, if it is in the tumor suppressor gene, it will interrupt the expression of the tumor suppressor genes. But they have also an ability to hijack the splicing donor acceptor process, and thereby they can activate oncogenes. So it allows you to study activation of oncogenes. and inactivation of tumor suppressor genes. And we managed these mice with mice that they have a fluxed allele of TGF1. This is not TIGF, but I'm sorry for this mistake. And so we crossed mice again with this recombinase that is under the CAG, which is ubiquitous, and this recombinase has a estrogen receptor that inactivates recombinase in the absence of tamoxifen. And when you add tamoxifen, what you will get activation of recombinase. And to recap, what happens after activation of recombinase, you have deletion of TGF and activation of sleeping beauty in the whole body. So you can see if TGF plays a role in all tissues. So the first tumor that we detected in these mice now is osteosarcoma. And this osteosarcoma developed only in mice that they have TGF deletion and SB or sleeping beauty activation. They don't develop at all in mice that they have only TGF deletion or sleeping beauty activation, indicating that TGFs can function as a tumor suppressor gene in osteosarcoma, provided that other genetic alterations contribute to this transformation. So, and yes, you can see this osteosarcoma, they have, I'm sorry, I don't know what happened. So that it metastasizes to the lung, and it is really very aggressive. So the other type of tumor that we detected in these mice is pancreatic cancer. You can see here, okay, the pancreas is very big, and it metastasizes to the lung. And you can see here with this white spot, and you can confirm just by H&E staining. And these tumors are pancreatic ductal adenocarcinoma that I will call PDAC from now and so on. And why they are PDAC? Because if you do immunostaining with this marker of PDAC CK19, you see that they are highly positive. Definitely they are PDAC. So I will focus now on this for the rest of my talk, just to give you like an overview about what is PDAC. Okay, it's really an extremely bad cancer. It can kill like in 2 weeks, and the problem is that it develops without any symptom. Really, I had a colleague a long time ago, and in France, I was really very sad to her about this news. So I heard that she had a pancreas, and she was my technician in the lab. So she had, they sent me an email that she developed, she was diagnosed with PDAC. 2 weeks later, okay, she passed away. And really another colleague, okay, she was a PI in my team. So I saw her husband in an airport, okay, because we were going to a conference together. He was healthy, big guy, everything was fine. Then we came back from the conference, and she said to me, her husband has some pain, back pain. And again, I think 10 days or so, he passed away. Just to tell you, this is an extremely aggressive type of cancer. So, and this is why we want to study, okay, hoping that maybe a day we can come up with some treatment, okay, to help patients. This is really my goal, the only goal, okay, for the rest of my career is to be able to do something for patients. So you can see here, survival is extremely bad, 7%. Now it is the fourth, but in 2023, it is expected to be the second. Risk factors, please pay attention to this alcohol and smoking. Really, this is the main cause of PDAC that people do not know, but pay please attention to it. Also, all these hamburgers and stuff like this with fat, please, it's very bad for PDAC, and also diabetes. So here, just to show the genetic alterations, it starts with KRAS. 90% of patients, they have mutations that activate KRAS. So this is the initiating event. But over time, these patients, they will develop other types of alterations in P53, P16, INK4, and SMAD4, and this will lead to the progression and demise of the patients. So back to our Sleeping Beauty mice. So we did some genomic profiling experiment using barcoded, because they have barcode this transposon, and we did whole genome sequencing. So, and we were able to identify alterations only in this gene, NF1, which encodes for a general inhibitor of RAS proteins. And you can see that KRAS, there is no alteration in the expression or also no mutation in KRAS. So the tumors that we have in the Sleeping Beauty mice did develop without any alteration in KRAS. So we have here an opportunity to identify the genetic alterations that drive PDAC in the fraction of patients that they do not have mutations in KRAS. The fraction is around 10%. So, and to study further NF1, we just interrogated, as all scientists do now, some public data sets, including the Cancer Genome Atlas, PACA Australia, PACA Canada, and Cosmic. And you see here, it's around 3.4% in the Cancer Genome Atlas, but it can reach 24.6% in the PACA Canada, which is extremely high. And so here, I listed all mutations that interact the function of NF1 as an inhibitor of RAS, and you can see half of the mutations inhibit or disable the function of NF1. And just this one diagram, you can see that very few samples, they have mutations in both NF1 and KRAS, but the vast majority of samples that they have mutation in NF1, they do not have mutations in KRAS, which suggests that maybe KRAS, inactivation of KRAS, can be an initiating event in pancreatic cancer. So we did also some immunohistochemistry, and we used a tissue microarray of 250 samples, which is very big. So we used a high specific antibody to NF1. And what we saw here, this is the normal tissue, the expression is there, is low, but it increases in a dramatic way in the perineoplastic lesions that we call pannings. Then it drops to the background level of in full PDAC when the cancer develops. So we have here a transient expression of NF1 during PDAC progression. Just if you quantify, you see it here very easy, normal tissue low, expression in penis very high, and in PDAC, again, it is very low. But so we did this immunofluorescence experiment with CK19. You see that they have pannings. All the samples that we analyzed, they have pannings. So those, they express both NF1 and CK19 very high, as we see here. But 18% of samples, they do not have any expression of NF1, which suggests that maybe other genetic alterations contribute to the loss of NF1 during PDAC progression. And we will discuss some of these genetic alterations. So to identify these genetic alterations, so we developed all these mouse model, a lot of work, trust me, okay, to generate these mice. So the first one, it has just the mutation KRAS G12D. The second one, we combine this KRAS G12D with homozygous deletion of P16. Here, homozygous deletion of SMED4 with KRAS G12D, and here, homozygous deletion of P53 with KRAS G12D. So we call this KC, KSC, and KSC and KPC. KSC here, the 3 mice, you can see normal is low, increase in penin, and it drops to the background level in PDAC. But KPC mice, you can see that the expression of INIF1 is very low. So which provide a hint, okay, that the expression of INIF1 might be regulated by P53. So I will discuss this later during my talk. So to study the role of NF1 in PDAC, so first we generated mice with pancreas-specific homozygous deletion of NF1 that you see here. So these mice, they were born at the expected Mendelian ratio, and they do not have any physiological or physical abnormalities. They were just normal. But analyzing these mice at the age of 6 months, what we saw is that deletion of NF1 can lead to the formation of this structure that we call acinar to ductal metaplasia, or IDM. And IDM is the first lesion found in patients with pancreatic cancer. And later, after 9 months and 12 months, you see that these mice develop these structures that we believe that they are penin structure. But these mice, despite developing neoplastic, perineoplastic lesions, they never developed PDAC. So they needs, okay, another genetic alteration to drive a PDAC. So just trying to find this genetic alteration that cooperate with NF1. So first we started just with KRAS, because 10% of patients, they have mutations in NF1 and KRAS. And so, so we were not able to get any mice that have homozygous deletion of NF1 and KRAS G12D, which indicate that this genetic combination is toxic. You cannot get it in mice. And to overcome this limitation, we decided just to study mice with heterozygous deletion of NF1 and KRAS G12D, just 1 copy of NF1 deleted. And you can see here the survival. These mice, they die extremely quickly, just 2 to 3 weeks after weaning, okay, separate them from their mother, they will develop aggressive PDAC, and they die very quickly. And these are PDACs, because you can see this here by CK19 and by another PDAC marker, MUC5AC. So the bottom line, okay, the take-home message is that deletion of 1 copy of NF1 is enough to drive a full-blown PDAC when KRAS G12D is combined with heterozygous deletion of NF1. I love this picture that I want to share with you. So this is very nice to see the result, the characterization in vitro, okay, of tumors. But here we want to do it just in vivo. And for this, we decided to use this mouse model that was generated by Dr. Sharpless. He was the NCI director. He stepped down last year. So, and in these mice, they put a firefly lysophase cDNA by knocking into the P16 INC4 locus, which makes the endogenous P16 ink promoter drives the expression of luciferase, but at the same time, it knocks out the endogenous P16 ink4. So there are homozygotes deleted for P16 ink4, but they express luciferase. And luciferase behaves exactly as endogenous P16 ink4. In that, you see it only during aging because P16 is a marker of aging, and you see it also when mice develop tumors like pancreatic cancer. So we generate again all these mice, okay, control mice that they have only KRAS G12D, mice that they have heterozygous deletion of NF1 only, and both alterations, and you see luciferase only in mice that they have the combined heterozygous deletion of NF1 and KRAS G12. This provides a very strong evidence that NF1 inactivation cooperates with KRAS to drive PDAC. So this is just an in vitro, very easy experiment to do compared to working with mice. This is a normal human epithelial cell line. And what is nice with this cell line is that if you overexpress KRAS, nothing is happening. And we took this cell line and we CRISPRed out NF1, 2 copies of NF1, nothing is happening. But if you combine NF1 deletion with KRAS G12D, what you see that these cells now deform spheres that they can go without attaching to the support, and they are highly motile, which means that they are invasive. And this tells us that inactivation of NF1 and KRAS G12D can induce cell transformation by a cell autonomous mechanism. It is not immunoseparation or something like that. It's a direct interaction between NF1 and KRAS G12D. Excuse me. So I showed to you earlier this slide with KC and KC that they show transient expression of NF1 in panels. So KPC mice, they do not have this transient expression of NF1, and even the expression of NF1 is very low, which this is why I suggest during the beginning of my talk that maybe P53 is regulating the expression of NF1. So we analyzed again the Cancer Genome Atlas, and we found that immunotumors that they have mutations in NF1, they have also mutations in P53. So they can be combined in the same samples. And this is the tissue microarray that we used before. I shared the video, so we have transient expression of NF1 in panels, and it is the background level at the level of PDAC. So P53 behaves the same. You can see increased expression in panel, but then it drops in PDAC, except that I would say 28% of samples, they have very, very high level of P53, which could be explained by the mutations. We know that PDAC has almost 50% of mutations that inactivate P53, but at the same time, destabilize P53. So this suggests that maybe P53, if regulating the expression of NF1, it does by a transcription-dependent mechanism. So it will be much more easier here because we took a human cell line, PDAC cell line MIA-PACA. We generated cells with doxycycline-inducible P53, and you can see that inducing the expression of P53 induces the expression of NF1 at the protein and the mRNA level. And so we observed the same phenomenon using another human P-Ductal line, PANC1. And here are the mutations of P53 that stabilize P53. And if you induce the expression of these mutations, you do not see an increase in the expression of NF1. You see it only when you induce the wild type P53, which confirms that P53 regulates the expression of NF1 by a transcription-dependent mechanism. So just trying to understand more this phenomenon. So we took the promoter of NF1 and we generate a lot of constructs with fragments, overlapping fragments of the promoter of NF1. And so we did a transfection with P53. You can see that P53 induces luciferase expression of NF1 only from this overlapping region here that I show in gray. Now, if you analyze more of this overlapping region, you do not see any P53 binding sequence. So which means that P53 regulates indirectly the expression of NF1, other factors that are involved. So to analyze this, so what we found a binding sequence for the transcription factor PML, and this other, that is less known, is TFA-P2C. By site-directed mutagenesis, if you kill this one, nothing is happening, but if you suppress the PML binding site, you block the induction of NF1 by P53. So PML plays a role in the induction of NF1 by P53. So to demonstrate this further, so we used cell lines that are wild type or deleted of PML. And so we did, again, luciferase assay. You can see that P53 induces the expression of NF1 only in wild type cells, but not in cells that they are deleted of PML. And if you do chromatin immunoprecipitation assay, you can see that P53 can bind to the NF1 promoter only in wild type cells. You can.
Sorry, can I ask you a quick question?
Yes.
In your experiment with P53 mutants, the two mutations that you generate are on the binding side of P53 to the DNA.
Yes.
So when you mutate the binding side of P53 DNA, you don't see expression of NF1. But if it needs an intermediary, then how you explain that those two mutations that are on the binding side are responsible for not seeing NF1?
So do you mean the mutations in P53, the one that we...
Yeah, there.
Yeah.
So those two mutations are on the DNA binding side of P53.
So these two mutations, in P53, these are mutations that inactivate P53.
P53 doesn't bind to DNA with these two mutations.
Cannot bind, to DNA,
So your conclusion was that P53 somehow contributes to the...
Yeah, it cannot bind to the promoter of NF1 and so cannot induce.
But you're saying that P53 doesn't really act on NF1 directly. It acts via your PML.
Exactly.
So then with these mutations, you should not see binding to PML, right?
Yeah, I totally agree with you, but we have not time to do this experiment, but I totally, this is logic, so I already said maybe that, yes, so P53 cannot bind to the NF1 promoter in cells that they are deleted of PML. And so this is in vivo using KC mice. You can see that PML can bind all the time in a constitutive manner to the NF1 promoter, but P53 can bind only in pannins. And please keep in mind that we saw increased expression of P53 only in pannins. So this is a good explanation as to why p53 is transiently expressed during PDAC progression. So now back to the genetics. So we want really to study, because this is a very strong interaction between p53 and NF1. Now, what is the role of this interaction in PDAC? But it was extremely hard to answer this question. So first, why? Because NF1 and p53, they are localized on the same chromosome. In patients, chromosome 70, and in mice on chromosome 11. But the genetic distance between NF1 and p53 is very small, is around 3 centimorgan, if you know what is a genetic distance, which makes it extremely hard to generate mice with the combined deletion of NF1 and p53. You keep crossing mice for years, and maybe you will get 1 or 2 mouse, out of 1,000 that they have both genetic inactivation. So, and doing some literature search, so we found this mouse model that we call mosaic analysis with double marker of MIDM. So this is the same mouse that you will see. It has cells that they do not have any staining. They have green cells that they are homozygous deletion of NF1 and p53, and red cells that they are just wild type for p53. So we started with these mice. So they have half of T.D. tomato and half of GFP in one chromosome 11, and mutated p53 and mutated NF1. The other chromosome has 11 has the other 2 halves of GFP and T.D. tomato, but they are wild type of p53 and NF1. So it's very complex, but just trust me that this is what will happen during cell division. You will have one cell that gives rise to 2 daughter cells, one green homozygous and one right. So in the same mouse, you have control, you have everything. You do not need to generate many mice to study this combination in cancer. So so we found that these mice develop pancreatic cancer. You can see here, this is a wild type mice, and this is mice with MIDM and NF1 and p53. You can see the tumor is very big, and it is yellow. Why it is yellow? Because it is expressing GFP. This is how you see it if you have a strong expression of GFP in the tumor. So CK19 positive, so PDAC. And you see here the survival. It is not as aggressive as KRAS, but still you see that these mice develop invasive PDAC. So now you can play, to see how it looks like the pancreas. So if you do a time course from 1 to 12 months, so what you see at the beginning in the pancreas, you see that you have green cells and you have red cells almost in equal number, because one cell mother can give rise to 2 cell daughters. It's logic to have the same cell number of green and red cells. But you see also some yellow cells. And these cells, they are in division because they have 4 chromosomes now. Before giving 2 chromosomes to one daughter cells, chromosome green, and the other red. And if you count over time, you see at the end, you do not see any red cells. You see just green cells that they develop. And if you quantify the gray to red ratio, you see a dramatic increase that reaches sometime 1,000 times 1,000 green cells per one red wild type cells. And this tells us also that this is a cell autonomous mechanism, because the tumor developed from cells that do have mutations in NF1 and P53. And so the metastasize to the lymph node and spleen and the liver, as you see here by green cells. And what I love with this MIDM is you can study the cancer formation, but you can also, it's very easy to study metastases in vitro, just doing GFP taken section and staining to see, just staining with DAPI and to identify green cells. So again, this is the normal epithelial cells. You do NF1 deletion, nothing happening, P53 deletion, nothing is happening. You combine the two, you have cell transformation. And the same with PML, because PML is between P53 and NF1. If you delete PML and NF1, you see cell transformation. So just to summarize what I showed to you, so far, so we found that NF1 is frequently mutated in PDAC. We detected transient expression of NF1 during PDAC progression. It is very high in panning. So we found that pancreas deletion of NF1 leads to IDM and sometimes pannings. If you delete one copy of NF1 in the background of KRAS G12D, so we have acceleration of PDAC. We saw also transient expression of P53, as we saw for NF1. And I showed to you that P53 regulates NF1 gene expression and doing so by PML. And you can see that if we inactivate P53 and NF1, it leads to formation of PDAC and by a cell autonomous mechanism. So to make it easy, so this is the graphical abstract that you can find with our paper that was published, I think, in November. So normal asina, you knock out NF1, you have IDM and panning, small panning. You express KRAS G12D, you have IDM and large panning. And if you add to that P53, either to NF1 or KRAS G12D, now you have formation of PDAC that metastasize to liver, lymph node, etc. And I hope that I convince you that now we have this 10% of patients that they do not have mutation in KRAS G12D. Now we might have an explanation for them why they developed the PDAC. And a lady with PDAC, she contacted me after she saw this paper, trying, like to work with me to see if she has a mutation in NF1 gene. Yes, so we are working, try to help her. At least we cannot propose any therapeutic approach now, but at least we can tell her if she has this mutation that drives a PDAC. So we started with TGIF, and just a few words very quickly about TGIF. We already published this work showing that TGIF deletion can lead to the formation of pancreatic cancer, PDAC. You see here, the survival, immunostainous CK19, and the size of the pancreas and the spleen, they develop splenomegaly. And they metastasize to several organs, and you see here liver, lung, lymph node. And so we use this nice mouse model with an inducible luciferase again that is specifically expressed in the pancreas. And very nicely, you can see here when you isolate the organs, you can see that you confirm the metastases to these organs. So we were very happy, but then we decided to develop mice with conditional overexpression of TGRF. And so what we saw, again, to our surprise, that if you overexpress TGF, these mice, they develop IDM and panins, like NF1 deletion. So you delete TGF, they develop PDAC. You overexpress TGF, they initiate PDAC. They do not progress to PDAC, but they initiate the transformation by the formation of IDM and PAN. So we decide to make these mice with KRAS G12D. And this is what we got, okay? So they have severe gout retardation, these mice. You can see they are very, very small. And even, okay, if you take a mouse that is as small as this one, a control mice, and you measure the weight of the muscle, you see a dramatic decrease in the weight of muscle, and also here the myofiber. This is how muscle looks like, which suggests that these mice did develop muscle cachexia. So we did many experiments. And just to summarize this RNA sequencing, so we found that the most upregulated genes are TGF-alpha and its receptor, EGFR, and the TWIC and its receptor, FN14. So to be honest, I am not a scientist guy. I am like a technical, okay. I love, okay, new techniques and all this, okay. And I have a strong background in like generating all types of constructs. And so I decided, okay, to take advantage of my expertise, and we start developing all these, okay, bispecific antibodies to like create new tools to block muscle cachexia. So it's very easy, but okay, you have to have the expertise. This is a monoclonal antibody for FN14, this one for TGF-alpha. If you have the sequence, you can combine them in all sorts of the configurations that we call BRCA1, BRCA2, and BRCA3. And in our hand, BRCA1 is the most powerful. So we would like to develop now clinical trials, okay, to see if these tools can block muscle cachexia in patients that they have pancreatic cancer or other types of cancer, like lung cancer, because they develop also muscle cachexia, and colon cancer also is a severe, okay, disease in terms of cachexia. And so this is what I have to share with you today. I hope that it was too long, to a lot of genetics, but I am very happy to take any question that you might have. And so you have my email address if you need any other information, like to understand better what I present to you today. Please just go ahead and shoot me an email, and I will be happy to answer your questions.
Thank you so much, Azeddine. Fantastic talk. Okay, we're going to take a few questions. If I may ask one quick question. So do you see like NF1 loss as a driver for other cancers that are dependent on KRAS mutation?
Yes. Say it again, please.
Sorry, do you see like NF1 loss as a driver for cancer progression in other cancers that are dependent on KRAS mutation? Or is it really specific to pancreatic cancer?
We saw this in only in pancreatic cancer, but I think that I saw a cancer cell paper. They found that NF1 inactivation can cooperate with KRAS, but not KRAS G12D, but KRAS G12D-C to drive lung cancer. But this, so this is not our mouse model, this MIDM. This one was developed by Dr. Lim in Stanford. And they used this mouse model to study glioblastoma. So with this genetic approach, MIDM and NF1 inactivation and P50, mice, if you do this in the brain using, for example, a nesting tree, they will develop severe glioblastoma.
Thank you. Yep,
Yes, sir.
Anthony?
So NF1 is associated also with neurofibroid. So is this just a context-dependent thing with different mutations in NF1? So do the
Yes.
individuals or do the mice develop neurofibroid tumor?
No, because these mice, all what we saw today it is pancreas specific. Yeah, if you delete NF1 in the pancreas, you will see the phenotype in the pancreas. You are not supposed to see it. Patients, we think
One mutations that you...
that they have NF1. This is a very good question. Really, I will try to understand. Yeah, because maybe, they develop other types of, because patients, they will not have mutation only in pancreatic cells. Maybe they will have a mutation in other cells if it is a somatic. But this also alteration could happen during cancer progression.
Yeah, so these are more, potentially there's somatic mutations post-transfer.
Yeah, exactly. And there is a phenomenon that happens in the pancreas. So keep in mind that they are on the chromosome 17, NF1 and P53. And we know that during PDAC at late stage, they develop a phenomenon that we call chromotripsis. So, and chromotripsis is that you have chromosome 17, you have pulverization of the whole chromosome, okay? Then cells, they need chromosome 17. They try to reconstitute the chromosome 17, but they do it in a random way. But many times, they lose both P53 and NF1 at the same time, because they reconstitute chromosome 17, but they do not do it in a perfect way. Maybe they lose fragments, and one of the fragments always has NF1 and P53. So it is during cancer progression.
Right, other question? Yeah, over there. Yep.
Thank you so much for the talk. So in humans, pannins are actually divided into 3 different types.
Yes.
Pannin 1 through pannin 3. I'm not sure whether there's a correlation in mice, but in humans we see pannin 1, 2, and 3, and you see a progression in dysplasia from low grade to high grade. And I'm wondering, as you talk about NF1 and pannins, whether you've been able to look at that and see what the expression of NF1 is. Do we see a drop in pannin 3 versus
Yes, we
NF1?
have this in our paper. You see a sharp increase in pannin 1. Then it starts to decrease at the level of pannin 2. And pannin 3 is almost like a full-blown PDAC that I showed to. It drops to the background level. It is very early, okay, this transient expression of NF1.
Right, any other questions? All right, is there any question on Zoom, online, Q&A? Yeah, all right, cool. Thank you so much again for coming here, visiting. Thank you
very much for
appreciate it.
coming.
Yeah.